This video is designed to highlight key aspects for the successful neural induction of human pluripotent stem cells using StemDiff neural induction medium. Neural induction is the first step in the differentiation of human pluripotent stem cells to neural progenitor cells, or NPCs, which can then be differentiated to mature cell types of the central nervous system. StemDiff neural induction medium is a defined, serum-free medium that is part of a complete system for the differentiation of HPSCs to neural progenitor cells. It rapidly and efficiently induces neural rosettes within 6 days, and highly enriched single-cell populations of NPCs are obtained within 12 days when used in conjunction with StemDiff neural rosette selection reagent and Agrawal 800 plates. Now let's examine the procedure in detail. It is critical to start with high-quality human pluripotent stem cell cultures in order to obtain highly efficient neural induction. Therefore, be sure to use cultures with less than 10% of the colonies displaying overt differentiation. Also, to start the neural induction process, cells should be harvested approximately 1 day prior to normal passaging to ensure they are in the growth phase. After removing the Agrawal 800 packaging, rinse each well to be used with 1 milliliter of DMEM-F12. Then add 0.5 milliliters of StemDiff neural induction medium supplemented with 10 micromolar Y27632 ROCK inhibitor. Centrifuge the plate at 2000 G for 5 minutes to remove any air bubbles from the microwells. Ensure the bubbles have been removed by observing the plate under the microscope. If bubbles are still present, simply centrifuge again at a higher speed for an additional 5 minutes. To prepare a single-cell suspension, aspirate the maintenance medium from the cell culture and rinse the cells once with 2 milliliters of DMEM-F12. Then add 3 milliliters of Accutase and incubate at 37 degrees Celsius until the cells easily detach from the plate with gentle shaking. This usually takes 5 to 10 minutes and should be verified microscopically. Next, gently pipette the cell suspension 2 to 3 times with a serological pipette to break up any clumps. Add 15 milliliters of DMEM-F12 to the cells and repeat pipetting up and down another 2 to 3 times. Then transfer the cells to a 15 milliliter or 50 milliliter conical tube. Rinse the plate with at least 5 milliliters of DMEM-F12 for every milliliter of Accutase used and add the rinsing solution to the tube. Centrifuge the cells at 300 G for 5 minutes. Then aspirate the supernatant and resuspend the pellet in 1 milliliter of StemDiff neural induction medium supplemented with 10 micromolar Y27632. Calculate the viable cell concentration using a 1 to 10 dilution sample of the resuspended cells in trypan blue and a hemocytometer. Because aggregate size affects differentiation outcome and the overall efficiency of neural induction, it is ideal to use the Agrawal system to produce uniformly sized embryoid bodies. For efficient neural induction in StemDiff neural induction medium, large neural aggregates in the range of 5 to 20,000 cells each should be formed using the Agrawal 800 plate. First, calculate the number of cells to be added to each well by multiplying the desired number of cells per aggregate by the number of microwells in one well. Then, add the calculated number of cells to each well and immediately pipette gently several times to distribute the cells evenly. Add StemDiff neural induction medium supplemented with 10 micromolar Y27632 to bring the final volume in each well to 2 milliliters. Pipette gently up and down to re-establish an even distribution of cells. Centrifuge the cells at 100 G for 3 minutes to capture the cells in the microwells. Then examine the plate under the microscope to verify the cells are evenly distributed among the microwells. Incubate the cells at 37 degrees Celsius for exactly 5 days with a daily medium change. After 24 hours, uniform neural aggregates will be visible. To change medium, carefully remove 1.5 milliliters of the spent medium. Be sure to keep the pipette tip toward the upper surface of the medium so as not to disturb the neural aggregates. Slowly add 1.5 milliliters of fresh medium by slightly touching the side of the well with the pipette tip at the surface of the medium. Use extra caution and do not drop medium into the well or create air bubbles, as both will disturb the neural aggregates. Then return the plate to 37 degrees Celsius and continue these 3/4 daily medium changes for 4 days. On day 5, harvest the neural aggregates by taking up the STEM-DIF neural induction medium from one agar well using a standard disposable 1 milliliter tip and then firmly releasing the medium back into the well. This will dislodge most of the aggregates. Make sure not to pipette the dislodged aggregates up and down with a regular 1 milliliter tip, but rather switch to a wide bore tip. Using the large bore tip, pipette 2 to 3 times inside the well, collect the aggregates, and then pass the suspension through a 37 micrometer reversible strainer to remove any single cells. Wash the AgarWell 800 surface 5 times each with 1 milliliter of DMEM-F12 and pass each of the washes over the same strainer. Remember to alternate each time between a standard and a large bore pipette tip. Afterwards, check the plate under the microscope to ensure that all the neural aggregates have been removed from the wells. If necessary, repeat the wash steps until they are removed. Finally, invert the 37 micrometer reversible strainer over a fresh conical tube and collect the neural aggregates by washing the strainer with 2 to 3 milliliters of STEM-DIF neural induction medium per well. Neural aggregates harvested from the AgarWell 800 plate should be plated whole onto PLO laminate coated plates. Plating small clusters or single cells can lead to cell death and thus low NPC yield later on. Transfer the neural aggregates from one well into a single well of a 6-well PLO laminate coated plate using a large bore pipette tip. Transfer the plate to the 37 degrees Celsius incubator and move it in quick but gentle side to side, forward to back motions to evenly distribute the neural aggregates. Culture the attached neural aggregates for 7 days at 37 degrees Celsius and perform a daily full medium change with STEM-DIF neural induction medium. Isolation of NPCs prior to the recommended 7 days may result in non-viable and uncommitted NPCs, which will not effectively differentiate into terminal cell types. Isolation of NPCs after the recommended 7 days can result in excessive neuronal growth. Morphological assessment and scoring of neural rosette formation provides a quantitative estimate of neural induction and should be performed on day 2 after plating. First, count all attached neural aggregates. Then, count only attached neural aggregates in which 50% or more of the area of each individual aggregate is filled with neural rosettes. The percentage of neural induction is calculated by number of neural aggregates with greater than or equal to 50% rosettes divided by the total number of neural aggregates times 100. These cultures show attached neural aggregates exhibiting 82% and 100% neural induction. A score of at least 75% is needed for successful isolation of a pure population of NPCs downstream. The flat cells, believed to be neural crest progenitor cells that often emerge alongside the neural rosettes, need to be removed from the culture. Failing to do so will lead to mixed populations of NPCs and neural crest cells, and downstream differentiation will be significantly affected. STEM-DIF neural rosette selection reagent has been specifically designed to allow selective detachment of neural rosette clusters while leaving the flat cells attached to the culture dish. Neural rosette clusters are then replated and cultured for a few days to allow CNS-type NPCs to grow out from the neural rosette clusters prior to subsequent single-cell passage of those NPCs. To remove flat cells, on day 7 of culture, aspirate the medium and wash with 1 milliliter of DMEM-F12. Add 1 milliliter of enzyme-free STEM-DIF neural rosette selection reagent and incubate for 1 hour at 37 degrees Celsius. Afterwards, carefully remove and discard the reagent using a micropipette outfitted with a disposable 1 milliliter tip. Using a new 1 milliliter tip, quickly expel 1 milliliter of DMEM-F12 onto the attached aggregates, specifically aiming for the rosette clusters. Without triturating, collect the neural rosette containing suspension into a 15 milliliter tube. Repeat this process until the majority of the neural rosettes appear to have been removed when viewed under a microscope. Next, centrifuge the 15 milliliter tube for 5 minutes at 350 G, aspirate off the supernatant and resuspend the rosette clusters in 2 milliliter of STEM-DIF neural induction medium by pipetting slowly up and down 1 or 2 times. The suspension should contain large clusters. Plate the neural rosette suspension derived from one well into one well of a fresh, pre-coated PLO laminate 6-well plate. Place the plate into a 37 degrees Celsius incubator and move it in quick but gentle side to side, forward to back motions to evenly distribute the rosette suspension. Culture the cells at 37 degrees Celsius with daily full medium changes using stem-diff neural induction medium until cells reach 80 to 90% confluence. Confluent cultures will consist of rosette-containing clusters and NPC outgrowths. Single-cell NPCs fill the gaps between these larger clusters. This typically takes between 2 to 5 days. The subsequent isolation of these single-cell populations of NPCs for downstream applications should then be optimized by the individual researcher for their particular application. For more information on stem-diff neural induction medium, please visit our website or email us at stemdiff@stemcell.com.